hope you can see me. Okay, it's 6, so we should start this session. And uh, First, let me introduce myself. My name is Shogo. I'm a second year PhD student at IISc Bangalore. Uh, I work with mostly genomics and different types of computational biology projects. I'll be your TA for this particular course on computational systems biology. And I was actually the TA for the same course last year as well. So, uh, if you have gone through the older TA videos and everything, I'll just let you know how the sessions work. So basically, it's a 2-hour session every week. For us, it's Tuesday on 6 to 8 p.m. Yeah, so it's Tuesdays 6 to 8 p.m. every week for this particular course, and I'll be taking the session. So for the 2 hours, for the first 1.5 hours, we'll go through the concepts covered in the week. I'll just quickly breeze through them and I try to explain those concepts from my perspective as well. And after that, in the last 1 hour or 45 minutes, we'll be solving some related assignment problems that will help you to do your original assignment every week. So I from for like getting to know me better. So I have a biotechnology background. I do not come from mathematics or physics. So I have a biotech background, and I've been doing computational biology for the last 4 5 years at least. And uh, yeah, so one thing I want to be very clear about is that I am a Python guy. I'm not a MATLAB guy. The MATLAB part, I won't be going deeper for many reasons. So sir has gotten the tutorial lectures for this in the NPTEL course only, so you can access that. I personally am not very well rehearsed with MATLAB, so I'll not touch that part. So yeah, so as I said, for the 2 hours, the first 1.5 hour, we'll be focusing on the concepts that has been covered, and in the last 45 minutes to 1 hour, we'll be focusing on the previous years or like related assignment questions. And all these sessions will be recorded, and this will be available on YouTube. And I think you will be able to access those videos and the PPT that I'm sharing as well. That will also be available uh, to you through the NPTEL team. I cannot directly give you the PPTs from here. So the NPTEL team will create a folder and everything, and I'll upload it there. And I think that Google Drive will be shared with you, all of you, so that you can access the lectures and like these problem solving sessions recordings and the slides that I will be showing. So I think yeah, that's about a good introduction as it gets. Like anyone wants to say anything, or anyone has any particular questions or uh, anything they want to ask, please go ahead and unmute yourself and do ask. Okay, I think there are no questions, so we'll get straight to the session. Uh, I hope you can see my screen. Okay. So, one thing before I start. So, I'll be assuming that you have come from mostly basic biology backgrounds, not much of mathematics or computation. So, if there is anyone who is coming from a mathematics or computational background, do let me know. Because I'll be more focusing on the biology and how to convert it into the mathematical terms and how to use that mathematics to understand the biological mechanisms of various things. So I'll not focus very, very deeply on the mathematics part, the modeling and everything. I will show you the modeling and how it is done and everything, but very nuanced parts of those, I'll not be focusing on those. Okay. So the name of the course is Computational Systems Biology. So last year also, when I started this course at the first lecture, I first wanted to give an idea about what is systems biology, what actually we mean by systems biology, and why is it important, and like a little bit of history about it. So, sorry, um, 1 second, I think there's some glitch. Yeah. So first, I'll suggest everyone this is a short paper that is freely available on the internet. You can just Google this up. Can a biologist fix a radio? I'm sure many of you have heard of this paper or read it prior to this, but this is actually a very interesting paper where this person, his name is Lezibnik. He started as an assistant professor in Cold Spring Harbor Laboratory. So when he first went there, and how he felt about different fields of biology, his field was apoptosis, and how he saw the field change from a very experimental focused field to systems biology, or a mathematical type of field. Uh, he pens that down very beautiful in this paper, and he tells us the requirement of a different language to understand biology. And in this case, the language will be mathematics or a computational language. But other than experimental biology, why we need to see biology as a system, not as a single entity. So little bit of context. So before the advent of this systems biology and everything, so the biological studies, were mostly focused on candidate genes or candidate pathways or candidate any particular thing in the cell that you want to check. Say, for example, you want to check a particular gene that is involved in a particular disease. So you try to knock that out, or you try to mutate that gene, and you see how the phenotype changes. So this was a very prevalent practice in days prior to systems biology. But what this branch of biology tells us is like you cannot see biology as a single or a biological system. Let's call a cell a biological system. So you cannot just see something inside the cell or inside the genome, and you cannot just treat it as a single entity. So you just cannot, say, for example, knock out that gene, as I was saying, and see what the disease or the phenotype is becoming. Because there's a reason why. So why is that? Because that gene or that particular area of the genome is not actually a single entity. It is controlled by various factors inside the cell, which we are calling as a system, or inside the genome, which is again a type of a system. So it is not exactly acting alone. There are factors which control its expression. There are factors which replace its expression. There are factors which tell it to transcribe or translate, or there is a various cascade of processes involved around that particular one single gene. So it is not acting in unison. So it's a system. So when you perturb that system, when you are trying to uh, like delete away that knock out that gene or anything, when you're perturbing that system, you're not only perturbing that particular um, gene, you're perturbing the whole system. So it's not exactly a good way to understand how the system or the cell or the genome is actually working. So I just gave you an example. You can read the paper and you'll understand what he's trying to say. It's a very simple paper. So I'll just leave a link here as well. So I think I have it open here somewhere. Yeah. Okay. Let's get back to this. Yeah. So I have made 5 points about the paper, what the paper is actually telling us. So the first one is the need for new languages. So as I was saying, ki biological methods focusing on individual components and their interactions, they are insufficient to understand the complex phenotypes. Say, for example, cancer. Cancer is not caused by a single gene or a single variation or anything like that. It's a multifactorial disease which is like which is contributed by a lot of genes, a lot of different things inside the cell. So it's not a single gene that you can identify and that gene is causing the cancer. So you somehow uh, uh, make therapies or something against it. So it's not as simple as that. So it's not a single entity. Or anything inside a cell or inside a living system is not a single entity. It's a whole system that is dependent upon each of the components of the system. So you perturb one component, a lot of other components get perturbed as well. So he raised the issue that we need new languages that can describe these properties arising from these interactions. And the languages that got developed are mostly mathematical ones. There are statistical nuances as well. But yeah, and the second important point was the importance of connections. So not only how these individual elements inside the system reside on the fact that how they are connected to each other, the how they are interacting with each other becomes very important. So these are not just individual parts; these are the connections between these individual parts is also important. Okay, and the third point is the limits of reductionism. So breaking down a complex system like a cell or a tissue or a particular organ or whatever you are trying to study inside a biological system, breaking down that complex system into its small parts can be helpful, and it can tell you about the individual functions of the individual parts. But in most cases, what will happen is you'll miss out the bigger picture. You're not seeing the system as a whole system. You're seeing the system as components of the system. So there are limits to this reductionism technique, and what we have been seeing it uh, for the experimental biology that has been going on from say 1970s to 1990s, early 2000s. Is like we take a particular component of the system and we try to study them while ignoring the whole system as a whole, how that particular component is having an impact on the whole system, and how connections to other components of the system from that particular element is actually driving the behavior of a particular system. So, uh, yeah. So, understanding the individual components is not enough. You need to look at the system as a whole, how it is behaving. And that is the point that is made by the value of systems thinking, and which basically tells you, okay, like the interactions are important. So these are essential in comprehending how the whole how like if a particular system is behaving in a certain way. Say for example a cancer cell, or if I can call it a system of cancer cells are behaving in a certain way, it's not because of one factor. It's a mixture of hundreds of factors which are interconnected, and which are like influencing each other to bring out that behavior, which we call as emergent properties. Emergent properties is something that you'll hear a lot around systems biology, because uh, mostly systems biology research that you see will be focused on understanding the mechanisms of different things inside the cell, which can be a signaling path, which can be a gene regulatory network, which can be a disease network or anything. So uh, like understanding the whole system becomes important. And the last and the most important point that he has made is the role of chance. So these complex systems, they are often influenced by chance events or random events as we call it. But how they respond to these events depend on their underlying structure and preparedness. So how they'll respond to this particular randomness will depend on the connections each of the element inside the system has with each other. So understanding this interplay between chance and preparedness, it's crucial. for predicting systems level behavior, which we are trying to do. So, which we are trying to predict how a particular system, in our case, a biological system, will work under certain conditions. That is uh, basically a very reductionist idea of systems biology. That is what systems biology tries to do. Okay, so the different language that we were talking about, in most cases, it's mathematics. So we try to understand different biological systems with the help of mathematical models, mathematical equations, and other things as well. But uh, we'll talk about mathematical models more because it's one of the most widely used uh, techniques to understand biological systems. So uh, this has been mathematical modeling has been there for decades, if not centuries. So it's nothing new. So we have tried to understand a lot of stuff around the world, be it physics, be it chemistry, using mathematical models, using certain equations that describe a particular behavior and putting them in a model to see how the whole behavior of a system changes by changing certain elements of the system. So this concept has been there for a very long time, but applying it to biology has not been very prevalent until like late 1990s and early 2000s. So uh, there are a few advantages of having of using mathematics to understand biology. The first thing is it's very precise. Uh, it's precise to that point. Like we are not that precise. So we do not know how precisely a biological system will work, but we have to design equations which will precisely describe the system. So it's kind of a paradox, but anyway, uh, it's very nuanced and very precise, and uh, it facilitates the formulation of ideas and identification of underlying assumptions. So we have a cell. Say for example, we have a cell. We want to model how that cell will take up glucose from its environment. So from our biological or domain knowledge, we find out that these are the different factors. Say for example, the the transport proteins in the cell, or the propensity of the cell to uptake a particular compound at a particular environment. We know all these biological phenomena. So we translate these biological phenomena into mathematical equations, mostly differential equations, and then we try to solve these equations to find at different points of time how the behavior of a system will change. And yeah, so since as you all know, biological systems are very complicated. A particular phenomenon inside this biological system that is going on will be very complicated. So you'll need a lot of equations, a lot of computation to understand actually what is going on. So you need a lot of computational power to do these numerical calculations, to solve these equations, to find out what you're trying to actually understand. So there are a few steps to do this kind of modeling, not in terms of like the technical steps, you write this equation or write that equation, which equation should you use. I'm not going into that technical detail right now. We will go into that details later in the later lectures. But this is just an overview of how and when to use this different mathematical models. The first and the most important thing when you are using any kind of models to describe something is that you have to know and identify the need. First, you have to define the question, what you are trying to answer, what is your model trying to address. That is the most and the first step and the most important step. If you can frame your question very well, very clearly, and very precisely, the model that you'll be building will be much more robust, much more uh, in line with the actual system. And if you have a very vague kind of a question and a very vague kind of a thing that you're trying to understand, it's going to be very hard to formulate this particular type of model. Next, hopefully you have understood what requirement you need. Now you move on to the next step is identifying these different types of parameters, different types of variables. So as I was saying, if you want to study how a cell is importing glucose from the environment, there will be certain factors involved. Say, for example, how the protein, how many glucose receptors are there in your cell, what is the rate of glucose like metabolism inside your cell, what is the requirement of glucose in your cell. So these kind of different factors actually govern uh, how a system will actually work. And these factors are like you can use these factors as proxy, as a parameter or a variable, different variables inside your equation. So using these variables and parameters, you can kind of mimic the different conditions inside your system, and you can use them appropriately. Third is the recognition of relevant data. So obviously you need data to first understand if there is a pattern inside the system or not. So understanding the data and understanding relevant patterns, and as there can be various ways you do this. You either do it as a way like you first collect the data and then you see if there are patterns, and then you try to formulate the question, and then you make a model and understand further. Or that it can be the other way around. You have an hypothesis, you make the model, and then you try to validate your model with existing data. So it can be both ways, but data is very important because that is what will give you a validation, or a concrete understanding what is happening inside the cell. Next, you have different physical principles that can be, say, for example, as I was saying, the requirement of glucose inside the cell. Say, for example, your cell already has a lot of glucose inside. Now, it will not go and again bring some glucose in because there will be hindrance inside the cell. So these kind of physical properties also govern how your system will work. So identifying these physical properties and incorporating them in your model is going to be very crucial to actually mimic the original system. And then you write your equations, then you solve those equations, you find some values and like, make a plot or make something else to understand actually how your system is changing with time. Uh, that is actually the final and the actually you can call the least important part of the whole modeling procedure. The most important are like the first and the most important thing is to have a proper question, a proper hypothesis that you want to ask from your model. Right. Okay. Moving on. So biological models, like applying mathematical models to biology. This is what I mean by biological models. So, so understanding these complex biological systems require more than characterizing individual molecules. It involves comprehending molecular interactions and pathways, as I was saying for this last 10-15 minutes. So it's important to consider everything that is possible inside biological systems. Now, these mathematical models play a crucial role in investigating how these regulatory processes are interconnected, how disruptions of these regulatory processes can lead to diseases. And these computational models will assist in systematically analyzing system perturbations, guiding experimental design, and evaluating molecules as therapeutic targets. As you know, like with the advent of AI and everything, so this has gone up a lot. Uh, Identifying molecules and as therapeutic targets. If you search this keyword in Google Scholar or somewhere, you'll find a list of Nature and Cell papers which deal with this. So it's kind of the talk of the current scenario right now. So yeah, and modeling is essential for biologists to analyze physiological processes and for pharmaceutical industry to support drug discovery and development. And the most recent advancement is omics. I'm sure a lot of you are familiar with omics, or at least you have heard the term omics. I personally work in genomics. I analyze genomic data and try to understand the different nuances that are present in the genome and how those can be functionally relevant uh, to like to kind of make a particular protein or not make a particular protein cause a disease, some things like that. So omics is omic, currently the scenario is omics and systems biology go hand in hand because the data I was talking about for validation, for understanding patterns, that data is primarily omics data currently because that's the best kind of biological data that you can have at the sequence level, at the transcriptomic level, at the protein level, at the amino acid level. You have access to this kind of data. So systems biology and omics are very, very closely connected, and yeah, one depends on the other. Okay, so this is a workflow about how. Oh, just a second, guys. I think I have an issue. Yeah, sorry for this. I don't know. There's something happening when I'm trying to uh, do the presentation. Yeah. So I hope you can see my screen. Please let me know if you can't. Okay. So this is kind of a workflow that is followed between for systems biology studies or mathematical study of biological systems. So you have a biological phenomenon that can be a disease, that can be a particular phenotype, that can be something else, whatever that can be. And you have some experimental data to validate it. So, for example, you have a disease, and you know that this disease is caused by a particular deficiency in a particular protein or a particular genetic defect inside the cell, but you want to find out if there are other factors affecting that particular phenotype or a disease. Now you take that experimental data, in most cases it will be omics data, and you kind of see and try to analyze that data and try to find patterns inside that data. Say, for example, you have a list of genes and you have expression values for all those genes uh, inside the cell, one for control group, one for a disease group. Now you compare different gene expression profiles, particular genes that are getting upregulated in your disease conditions, particular genes that are getting downregulated in your disease conditions, things like that. You try to find these particular patterns, and then you formulate an hypothesis. Okay, I think this particular genes might be interacting with each other or doing something, and that is bringing up this particular phenotype. So you make that particular model, you do that model, and you test that hypothesis on your experimental data. See if your model can actually explain your data or not. If yes, good, you do a wet lab experiment to validate it, and if not, then you try to find new data or try to find a new hypothesis, and you try to do this like over and over again until you find a right hypothesis and the right model that can explain your data. Now, this is a standard workflow that is followed in most labs. So in big labs, you'll find there are site for computation. Sorry. There's a place for computation and stuff, in silico stuff, and there is a place for your wet lab stuff. So most molecular biology labs now you will see like the people are doing computational studies to generate a hypothesis or mm, get an idea about a system, and then try to validate that on some sort of wet lab technique using PCR and stuff like that. So this is a very interconnected uh, area where wet lab experiments can actually give you hypothesis about how the system is working, and then you can make a model out of it. Or it can be the other way around. You have an hypothesis, you make a model, and you see the data is getting validated. Now you want to validate it using wet lab again. So in both ways it can work. So just to quickly go through it, biological systems with numerous molecular components exhibit these emergent properties. I said you'll come across this term emergent properties a lot. So yeah, and nonlinear reaction kinetics and complexities like cell structure contribute to unpredictable behavior. And the idea of these mathematical models is to capture all these complexities and allowing to do in silico or computer simulations of these particular models and try to mimic that system. And theoretical models tested experimentally, guide adjustments in framework or experiment setups, and simulations. Uh, and models predict systems behavior and systematically investigate perturbations induced by these various factors. And the combination of these dry and wet lab approaches in systems biology aids in hypothesis development. And there are a set of applications to this. Metabolic engineering is one major application. We'll talk about it in, I think, week 8 or week 9. We have a whole module on metabolic engineering, and we'll talk about it then only. So I'll skip this. Now, when we talk about a system, we also have to talk about the stability of a system. Like if that system is actually a stable one or an unstable one, can it keep on doing what it is doing uh, in the future as well, or is it like it's not an unstable one, it's an unstable system, it can break down anytime. So this is a very simple concept of steady state. So these are these are fixed points, or these are also known as equilibrium points, where the rate of change of a particular parameter, so for example, it can be the rate of change of production of a particular protein, it can be the rate of change of, say, for example, expression of a gene, or anything. It can be, depending on what question you're trying to answer, it can be anything. But the idea is that parameter will not change. It's the change of that parameter, which is given by the dNdt term here, is actually zero. In simpler terms, it's where the system isn't changing over time. So uh, you have a system which is stable, which does not change over time. Now, this stability can be of two types, which is intuitive. So this is on the left is a diagram that actually shows different types of stability. So this, you have a stability here. These all three are steady states actually. In all three cases, it's the system is not moving. But you classify stable states based on predicting whether a system will move if a particular perturbation occurs or not. Say, for example, in this case, the S1 ball here, if you just push it slightly to the right or to the left, it's most likely going to come back to its own system, own state. So this is a steady state, but it's a stable steady state. If you move the system out of that steady state slightly, it has a attraction to come back to that particular steady state. Yeah, and you have steady states like this in this case U1, which is basically it's steady, it's not moving, but if you push it on either side, on to the left or the right, it will not come back naturally to its current position. It will roll away further down. So this is actually, although it's a it's a steady state, it's unstable. So it's called as unstable equilibrium, and this is known as stable equilibrium. So as I said, if a small disturbance away from the steady state causes the system to return to the steady state over time, then it's called stable. If it does not return to the steady state uh, after pushing it slightly away, it it's unstable. And there is another concept of attractiveness. So basically, if the perturbation leads the system to fall back to the steady state, then the steady state is attractive. So for example, this will be attractive because this, if you push it higher up, it will come back to here. So it's attractive, and the other one is repulsive in this case here. So if you push it left or right, it will not come back here. There is no attraction of that state for that particular system. It's quite simple. When you're defining a system, the system will have different states, and will in most of the cases will be concerned with steady states because of this particular beautiful condition where d and dt equal to zero. So the idea is you write a lot of mathematical equations, a lot of these differential equations and everything, and you analyze the system when the system is in steady state, when this particular condition will hold d and dt equal to zero. So we'll see how we do that in future time, but this is generally how it is done. Okay. So after steady states and different types of steady state, we have another a few concepts of different systems. So one is robustness, the other is resilience. So robustness is basically the ability of a system to remain or reach the same stable steady state, and resilience is the ability of this system to return to a previous steady state or establish a new state after significant perturbations. So I have given certain examples. I know the concept is a little bit confusing, so that I have given three, four examples here. So for example, you have a plant. So when if a plant is robust but not resilient, it will only grow under light conditions, but will struggle switching between these light conditions. It's robust. It can grow under a particular light condition. It will grow very beautifully. But if you put it away in another light condition, it will not grow that much. So it's robust, but it's not resilient. Another example is being resilient and not robust. So for example, the plant becomes dormant in the dark, but restores the growth rapidly in desired light conditions. So it kind of adapts to the environment. When it is dark, it will not grow. When there is light, it will grow. So that concept is resilience. And if you combine both of them, so it's robust and resilient, it will grow under all the conditions. And if it's neither robust nor resilient, it'll only grow under a particular light condition and it'll die if that condition is not met. So I think these examples will help you understand what robustness and resilience means. These definitions, these terms I'm saying because uh, when you study systems biology, when you try to see a particular thing as a system, there are certain nuances with the system. There are certain parameters of a system that we define the system with. So robustness, resilience, steady states, all of these are actually parameters of a system by which we define a system. So we'll come across these terms later in the lectures, but yeah, I just wanted to give you an idea about what these different kind of things mean. So coming to that part of the talk where I was talking about different types of models and how you can connect it with the data, and you can take assumptions from the data and connect it back to the model. So this interplay between the data and the model. So here the here one important concept is the overfitting and the underfitting of the data. So do not go into all this like mathematical uh, uh, stuff. I've just written it to kind of show you what is the mathematical formulation for expected loss, or when you're calculating some sort of uh, RMAC or something like that. You're calculating some sort of loss. So the loss actually, so for example, you have a model. You simulate a model, you get a certain type of behavior. By behavior, I mean a curve, uh, change of the system over time. That is technically behavior of a system means that change of the system over time. So you create a model, you simulate that model, and you get a particular behavior. Now you have another data where you have the actual behavior of the system, and there will be a difference between those, because like obviously your math model will not be perfect. It cannot perfectly predict everything. There will be differences, so that difference we calculate as the expected loss from a model. So this formula is given by, like, this expected loss is actually your noise, your bias, and your variance. There are three components to this. If we just forget about noise for a moment, because it's inherent of the data. So bias and variance, these two things will define if your model is overfitting or underfitting. So what are these concepts of overfitting and underfitting? So for example, this is a data. These are data points. So the dots are the data points, and the curve is the curve that actually explains the behavior of this data points. So this particular uh, plot on the top is actually showing overfitting of the data. So you have a data set. You're trying to fit a curve through that data. If the curve actually fits every point of the data, say for example you have a data set, you are trying to fit a polynomial curve to it. Now if the curve is fitting every point in the data, that means something is wrong, because you do not want actually a curve for that particular data set. You want a curve for the whole system. some generalized curve that can explain the whole system, not that particular data point. So if that happens, that is what is known as overfitting. In this case, your variance will be very high. And if you have a, a, in a case where your bias is too high, so your data will underfit. Basically, your model or your curve will not be able to explain the data properly. So there are two cases where your model or your curve explains your data only, and it explains nothing else. It just explains the different points, nothing else. No explanation about the system. That is overfitting. And in the opposite end of the spectrum, in the opposite case, where your data cannot be fit into, sorry, your curve cannot be fit into your data, so your curve cannot explain that data anyhow. So that is underfitting. So this is actually on the right is a bias variance plot. So on the this is basically on your x-axis is your model complexity and your y-axis is your error, and the total error is given by this black curve, and uh, the blue curve and the red curve are showing bias, variance and bias respectively. So actually, if you lower your bias, if you I don't like if you go if you say for example bias tends to 0, your variance will be very high. On the other hand, if your variance is too low, your bias will be very high. So you do not want either of these extreme conditions. You want something in between where your variance and your bias are kind of similar. And in that case only, if you see the total error curve, it's actually the lowest error in this particular case where your bias and variance are balanced and it's almost equal. So um, actually example, I think this is an example only. So I'll explain this particular plot. Just give me a minute. Oh, yeah. Okay, let me explain this. So in the first panel, you have a few set of data points, and on this top, if you see this expression, so these are actually polynomial curves that I'm trying to fit into the data. So you have, so if I'm trying to fit, say, 1x, x square, and x cube, till x cube, I'm trying to fit that particular model into my data. So you see, it explains actually pretty well. So this is your pink is your data curve. So basically the model that is fit to your data, and the blue one is actually the model that is actually explaining your system. So in this case, it's not that bad. See, the data point and the model behavior are not way apart. So if this happens, if you go up till x cube, so this actually the top panel is showing you a good fit of the model. All three, these are different data sets and three different distributions, and in all three you have fit a model, a polynomial model that goes up to x cube. Now, if you do something like this, so if you want to fit, you say ki like x cube is okay, but I want to go further ahead in the polynomial curve. So I want to explain further. So you go up to say x to the power 14. Now what you see is the pink curve is actually fitting all the data points. It is just going off from the actual behavior and just going after the data points and fitting the data points. It's just only fitting the data points. It's not telling you the actual behavior of the system, which is actually given by this blue line. This is the original behavior, and this is your predicted. The pink line is your predicted behavior. So if you increase your number of polynomials that you're fitting, what will happen is it will go and fit to the data itself, and it will not explain any kind of systems level behavior. Same is the case for other two. So this is a case where basically your data is overfitting, your variance is too high. So your curve passes through each and every data point if you notice. But each and every data point is not actually telling you the full story of the system. The system story will be told by the whole set of data points and the best curve that fits the data. In this case, since you have fitted a very highly like a high polynomial model here, what it is doing is it's explaining your data points, but it's not actually explaining the actual behavior of the system. Now, if you look at this blue line is actually does not fit into any of the data points. It's actually passing around the data points here and there. So that is what we want. That because my data points will not exactly tell me my system, right? It's a version of my system. So for example, I'm taking gene expression data. I When and how I have taken this data will uh, give me a particular curve, but that doesn't mean the system will behave like that throughout everything. You need a generalized expression. You need a generalized curve that explains your whole system. In this case, your blue line will explain it much better than this red line, because your red line has overfitted in your model. It's just going through each and every data point and it's fitting the curve in that, which is not what you want. You want to have a systems level understanding of what is going on inside your system through the data, and that doesn't necessarily mean that you have to fit every data point to your curve. So actually, I hope you have understood this concept. I do not have better examples currently. This is the example that I put. So this example is actually of a overfitting model. In the underfitting model, your curve will be nowhere near the data points. It will be somewhere way off. It will be even weirder than this. So that is what I was explaining. Ki your variance and bias should be balanced somehow, somewhere where your error will be the lowest. So that is the point that you have to optimize. So we'll be optimizing different models in future classes and so on. So we will optimize that model to have a good balance of variance and bias, because if you have either of them very high, your model will not work properly. I hope I've been able to make you understand this. Okay. Okay, so after this, I'll move into a couple of examples and I'll show you how actually these different types of models work. So this is actually a very interesting model. This is one of the earliest mathematical models in biology. I think this is from 1950s. So, and the beautiful thing about this is this still holds till date, till 2025. Uh, we are taught gate model by Hodgkin and Huxley, and this model still holds to explain when and how a neuron will open its channel and close its channels. It's actually a very beautiful piece of science that was that we got in like 70 years ago, but it still stands. It's one of the very few things in science that has stood the test of time. You can go and read about it. There are a lot of uh, books and papers around this particular model. So anyway, so this is actually a model by Hodgkin and Huxley, sorry, which explains the opening and closing of different ion channels inside the neuron. So what happens is ion channels in the membrane of the neuron undergo conformational changes in response to a variation in electric field. So the opening and closing is controlled by the electric field around the neuron. So that is very important. This conformation change of when electric field changes causes the channel to move from open to closed states or vice versa. So this change in electric field is what controls when the channel will open and when different ions will go inside, and when the channel will close and no ions can go outside or inside. The most important thing is this: because of this opening and closing of these channels and flowing of the ions from inside to outside and outside to inside, uh, the concept of potential comes up. So when a neuron is firing signals, it's actually opening and closing its ion channels and changing electric field, and so electrons are moving in a particular direction. So neuronal signals are fully controlled by how and when this particular ion channels are opening and closing. So here is basically, I'll not go into the details very much, but I just want to show you what this figure means. So you have a non you have a permissive state which is on the right, and you have a non you have a sorry yeah we have a permissive state which is on the left, and you have a non-permissive state on the right. So your electric field we move from your non-permissive to your permissive state, and you have so probability of being in this permissive state will be y, and probability being not being in that permissive state will be 1 minus y. So Hodgkin and Huxley did not know about the ions and different ions that are flowing in and out. What they assumed were there were gating particles. There are particles inside the neuron which actually controls this particular flow. So they did not know much about ions. What they conceptualized is about gating particles. So your non-permissive state, your y-axis is your energy actually. Your non-permissive state is actually at a higher energy state than your permissive state. But as we saw as we have seen in chemical kinetics as well, if you want to move from one state to the other state, you have to cross an energy barrier. This is the energy barrier that you have to cross and go to the other side. Now, this energy barrier in biochemical reactions, if you recall, this energy barrier is lowered when you have an enzyme working with it. In this case, this energy barrier is lowered when you have electric field around the ion channels. So this this conversion of going from one state to other state and another state to back to this state is given by this particular reaction. So you go from A to B with a rate of beta, and you come back from B to A with a rate of alpha. So this delta Z FV that you see, this is the difference in energy levels when there is a particular electric field that is or a voltage potential that is around the particular neuron. So if you want to model this particular thing, this whole phenomenon, so you model the rates of this particular opening and closing. So going from permissive to non-permissive, non-permissive to permissive. So uh, if you want to model that, there are very good equations available to model this particular rate of reactions. If you notice this is a very familiar equation, A to the power minus EA by RT, if you remember from class 12 anyway. So alpha and beta can be modeled like this. And the second part of the model is basically the reaction between the opening and the closed states. It's a first order chemical kinetic reaction, so which is given by this equation. So, and at steady state, why I was talking about steady state and different kind of things like that. So, we always calculate this variables, or always try to see the model how the model is behaving at steady state, because your change with time will be zero. So at your steady state, your dy dt will be zero, and if you equate this, you get a particular equation, and if you plot it, you get a behavior of how alpha and beta are changing with time. So this is actually a very interesting phenomena that happens inside neurons, and this model actually, as I was saying, like, still holds the test of time. And this so many advances in science, but still this model is able to explain opening and closing of ion channels and uh, other things inside the neuron. So this is one example that I wanted to show you. Yeah, if you are interested, please go and read this particular model. You'll find it in n number of neuroscience textbooks or internet or YouTube. Anywhere you will find this model. It's very interesting. Another model that I want to talk about is basically a signaling model, signaling pathway model. So, in a lot of cases, you'll see the systems biology has been applied to signaling pathways, various networks, primarily regulation networks or transcription regulated networks, things like that, trying to understand what will happen if I perturb a particular uh, element inside the system. So in this case, the system is a signaling pathway. So in a signaling pathway, we'll have a set of proteins and a set of molecules which are controlled by a set of genes, and those things are working in unison. So there will be one product which will create another product, which will create another product, and downstream like this, the whole signaling pathway is getting formed. Now, one such example of a signaling pathway is NF-kappa B. So NF-kappa B is actually a transcription factor. So what it does is it goes inside the nucleus and it binds to the DNA, and it controls or regulates the expression of other genes. So this is a very this is a very, like, what to say, very commonly found transcription factor, commonly studied as well. And it's a protein that plays a role in many important processes, including inflammation, immunity, cell growth. So NF-kappa B, by the way, is the transcription factor that gets activated when you have fever. So it's a very widely used transcription factor. So it's normally found in the cytoplasm, but since it's a transcription factor, it must move to the nucleus and bind to the DNA to regulate expression of different genes. So uh, there is a mechanism in place to allow these regulations to go inside and bind to the DNA and allow this regulation. There's a very sophisticated mechanism in place. So to prevent, initially when it is inside the cytoplasm, NF-kappa B, to prevent that NF-kappa B to moving to the nucleus inappropriately, it is normally bound to inhibitory proteins which are known as I-kappa Bs. So normally in your free state, when the NF-kappa B is in the cytoplasm, it is bound to another protein which does not allow it to move into the nucleus. Now, when a signal comes and tells that this NF-kappa B needs to be activated for some reason, so what happens is the other protein, the I-kappa Bs, they are degraded instantaneously. Now these NF-kappa Bs are free and they can go inside the nucleus. And conversely, one of the genes that NF-kappa B turns on actually codes for that particular protein, which is the I-kappa B, which was actually not allowing NF-kappa B to go inside. So what happens is, like, NF-kappa B goes inside, it turns on certain number of genes, one of them being this I-kappa B. So enough amount of I-kappa B, sorry, gets formed, and what happens is now NF-kappa B binds to the I-kappa B and now cannot go back inside. So it's actually a feedback loop. It's actually a negative feedback loop. So you have two proteins, you degrade one protein and move the other protein inside the nucleus. That protein actually helps code for the other protein. So if that NF-kappa B is inside the nucleus for a very long time, it will produce a lot of I-kappa Bs, which in turn will inhibit NF-kappa B. So this is a regulatory network, but specifically this is a negative feedback loop. So the cycle of activation and inhibition helps to keep NF-kappa B, amount of NF-kappa B inside the cell, in check. So there are mathematical models which describe this particular phenomena. This is actually a very interesting phenomena. And this on the right is actually the model that uh, explains the different phenomena of I kappa B and NF kappa B, the dynamics between those. I'll not go into the details of the model. There are a lot of variables involved here, but I just wanted to give an idea about how different processes can be modeled. Right. So this kind of signaling pathways, this kind of regulatory network, or anything like that, everything can be modeled. And so this is basically the model which takes into account different things inside the cell, so different behaviors and everything. And this is how actually the curves turn out. So I am not sure if you're able to see it. So this is how actually the behavior of NF kappa B over time turns out, and this is actually the network. Uh, TNF on top, if you see, TNF is a it's actually a cytokine, which is tumor necrosis factor. It actually comes and activates NF kappa B and degrades I kappa B. So yeah, different levels of regulation is present. So the more complex your model becomes, the more regulatory elements you are being able to add to that particular model. Okay, so yeah, I hope I have been able to give you an overview about systems biology, why we should be studying systems biology, and why we should be applying systems biology to various biological systems, because it's actually interesting and it's actually much easier to do at least the mathematical part or the statistical part, computational part is much easier to do than a lab experiment. So this slide actually sums up systems biology in general. So on the left hand side you have this. So basically your DNA. From DNA you create the RNA, then you create the protein. From the protein you create metabolites, and you study the function of different metabolites and what happens when a particular phenotype is produced, and what are the different proteins and the metabolites and genes involved in that particular phenotype. So from the left to the right, so till from genomics to transcriptomics. So genomics is the study of the gene, DNA. Transcriptomics is the study of the RNA, proteomics for protein. Network biology is the interaction between these transcriptions, transcriptomics, metabolomics, genomics, everything. Is It comes under the field of network biology. Then you have like how these different interactions make up metabolites or uh, different elements. Sorry, different elements in the cell, and uh, then all this data, this left part till systems biology is all data, right? So all this data you have collected, now you want to see emergent properties out of this particular system. So you want to see how this particular system is behaving according to the data. You want to fit a curve to the data to explain how the system is behaving. So there comes the systems biology part, so which is this part, and it generally comes from left to right, but as I said, it can go from right to left as well. Uh, Bring in a hypothesis from your different models and everything, and you test that hypothesis using omics data, genomics, proteomics, transcriptomics, anything. So this slide actually is taken from a Cell paper. I forgot the name of the paper, by the way. It was taken last time only, so I've forgotten which is the name of the paper. If you want to go and see the paper, you can just do a reverse Google search of this particular image, and I think you'll get the paper. I do not have it currently with me. I've forgotten which one it is. So, in that paper actually explains the different fields and how they come together with this different type of omics technologies, different types of mathematical technology techniques and systems biology, how it comes together to kind of answer certain hypotheses, answer certain questions about the system. So yeah, this till this part is just covering up. So today's first hour it was a little different. From the next week, we'll cover exactly what Sir is teaching. This week, I wanted to give you a little bit more overview, more than what, like a different perspective to what Sir has said. And I think I have given you that. And if you have any questions, you can ask me, or else we'll move ahead with solving few assignment related questions. Okay, then. We'll go into the problem solving part. So I'll keep the question on the screen for about a minute or two, depending on the length of the question. And after that, we'll discuss the answers. Okay. So let's go for the first one. These are very, like, some of the questions are very simple and uh, like it's based on understanding of what is systems biology and what is mathematical modeling, these kind of things. The question will get a little bit more trickier in the next classes. This is just the first and the introduction class. So yeah. Okay. The question is which of the following is true for a mathematical model? So these are the answers. So firstly, all models are practically wrong. So because you cannot expect to kind of translate such a complex system into like 5 or 6 different equations. It's never possible. So there will be parts where you're always missing out, and technically your model will always be wrong. How wrong will depend on you basically. How close you can make the model to an actual system will be your expertise. But other than that, even if the best model that is there that is explaining the system, that will also be wrong on certain levels. So technically, all models are wrong. And it's only true for the assumptions that we make. So our model will only tell us about the system depending on the assumptions that we make about the model. If we make correct assumptions, we'll get results closer to the original system. If we don't make correct assumptions, we'll get wayward answers. So that is there as well. And this is actually false. If you even if you consider large data set, if your variance is very high, you will overfit your data. So there is uh, no correlation between this uh, size of the data and overfitting or underfitting the data. It's not correlated like that. And it's in most cases, complex model will always perform better than a simple model, which is intuitive. But there are caveats to this. So if you look through literature enough, there has been cases where complex models have not been able to explain certain behavior, whereas simple model has been able to. So there are nuances to this. I'll not take this at face value, but yeah. And yeah, it's simple models. They may be preferable under certain conditions. So yeah, moving on. You can write the answer in the chat box if you want. Okay. I don't think we have the answer. I'll move quickly over this because we have better questions to move on to. Anyway, so the point is that you are told to build a model that will predict the temperature of a particular city. Okay, I'll discuss. Yeah. Uh, yeah. Uh, yeah, you're asked to make a model to predict the temperature of a particular city, and you have been given data for 15 years. So it'll it has measured different parameters, which include humidity, velocity, air velocity, pressure, etc. And you are using a polynomial regression model, and you are optimizing it using RMSE. So firstly, the model that you build is mathematical. First and foremost, because you're predicting a particular thing using different parameters of a mod of different factors using a mathematical model. So the model is mathematical firstly. Secondly, it's deterministic. Why is it deterministic? Because there is no randomness associated with the model. So if you have a, say for example, you're using a regression model, and uh, uh, and you like use another term which actually uh, accounts for the noise in the data. So then that will be that you're incorporating a random parameter inside the model. So in that case, it will not be deterministic. So what does deterministic mean is if you give a set of input to the model, will it give the same output every time or not? So if there is a randomness associated in the model, so every every time you give the same input, it will not give you the same output. But if you if your model is deterministic, it does not have any randomness. The same input every time will give you the same output. And it's empirical because you started like you are modeling based on real world data. So you are seeing the data, you're trying to find patterns inside the data, and then building a framework which will explain the data. That is what empirical modeling means actually. So yeah, technically we are doing mathematical, deterministic, and empirical modeling. So yeah, I think that answer was correct actually. Whoever gave it, it's not stochastic, by the way. So it's not stochastic. Stochastic models we will see. Actually, uh, I think we have a module on stochastic model if I'm not wrong. But here also like a set of random variables or random parameters are involved as well. So it's not stochastic. It's actually deterministic. So yeah. Okay. Do we have any answers? Stable complex. Okay. Okay. Yeah, it's correct. So biological systems are actually very robust and resilient both. In this case, the first option is sensitive, the opposite to that will be resilient. So I was talking about robustness and resilience because of the fact because biological systems are very robust and resilient. So if you just try to push it away from a particular state, it's most like if it's a stable state, it's most likely going to come back. So it's very stable and very uh, non-sensitive, robust and resilient. Consistent, not so much because like there is a lot of heterogeneity involved. The third and the fifth options are actually very similar because if you have a lot of heterogeneity involved in a system, it's not going to be very consistent across the system. So it's not homogeneous, it's not consistent, and obviously it's complex because there are a lot of parameters and variables involved. So yeah. Oh, sorry. Okay. I'll go to the answers. So, someone wrote in the last option. Actually, the last option is incorrect. If you go and look up, there are different models where of Michaelis-Menten when there are inhibitors present, when there are enhancers present, and when there are other types of intermediary products present. So it's not the same when there are inhibitors present. It will not remain the same. There are separate equations for Michaelis-Menten if you look it up. So yeah, fourth option is not correct. The correct option is a low value of Km indicates the binding of the enzyme with the substrate is high. Why is it? Because if you recall, I just quickly, I think I can show you here. Yeah, actually, this is the Michaelis-Menten equation. If you remember, V equal to K max, V equal to V max into S by Km plus S. So if you have a lower Km, your V will be higher. So the velocity of the reaction will be higher if you have a lower Km value, which is your Michaelis-Menten constant. So in this case, so, just a second. Yeah. In this case, what will happen is if your Km value is low, your velocity will increase, which basically indicates that your enzyme and substrate is binding at a very high rate. So, I don't think- okay, let's move on to the next one. So these are simple computational MCQs, if you can call them, just to revise the concepts of loops and things like that. Very basic stuff. So yeah, it's actually MATLAB, so I'll not go into very details of this, but yeah, you will get questions like these, but like simple only. It will not be very complicated. You just need to, if you're not familiar with MATLAB, I'll suggest you take the tutorial sessions that are like see the tutorial sessions that are there in the course itself, and also like knowing the syntax would be enough because MATLAB is very similar to Python. So if you know Python, uh, MATLAB will not be very hard to understand. You just need to know the syntax. That's all. Okay. The answer is two because it's 3 to 4. It considers 3 and 4 as well. Uh, and the question is the number of iterations. It's not the value of t actually. So it'll go twice, one for 3, one for 4. So the number of loops will be 2. It's simple. If you are asked the value of p, then you have to add up these and uh, tell the value of p. But this is the number of loops they're asking. Okay. Do we have any answers? So, Okay, so it's actually a very simple question. So the question is you have a reaction where you have an enzyme and a substrate which is forming an enzyme substrate complex which is breaking down to form enzyme plus product. Uh, a similar type of equations may seen earlier for Michaelis Menten kinetics, but one subtle difference is there. The enzyme substrate complex breaking down to form enzyme and product, that's not a unidirectional reaction. In this case, it's a bidirectional reaction. In most cases, what will happen is you form enzyme and substrate, you add enzyme and substrate to form the enzyme substrate complex, which is actually uh, that reaction is reversible, but the formation of product from that enzyme substrate complex is actually irreversible. But in this case, it's reversible. So if your velocity of the reaction is at the highest, which is V max, what it means is there's a lot of substrates present because your direction of the reaction is going uh, rightward because your product is forming. So you have a lot of substrates present, and your ES complex will be at the very highest point because all these are reversible reactions. Now, if you consider if this particular reaction was irreversible, your ES concentration would have been very low because the moment ES is getting formed, it will be converting into EA plus P. But in this case, since this is reversible, so your ES will be at a highest point, and since the reaction is at a maximum, you need your substrates to be very abundant. So yeah, okay, moving on to the next one. Okay, guys, I'll be back in just a minute. I'll just have some water and come back. Can you hear me now? Okay. Sorry for that. Did I miss the last question? I just go over it again. So basically, this is a system where all the elements are uh, inhibiting each other or down regulating each other. So that's the reason it's called reprocessor. In oscillation, what will happen is there will be some elements which will uh, upregulate some elements, and there will be some elements which will downregulate it. So the system will go from top to bottom like this, peak to trough. So that will be an oscillation. We'll see examples of oscillation, amplification, toggle switch, all of these in the later parts where we talk about networks and how networks behave. But actually, yeah, reprocessor is something that is widely found in biological systems. This is actually very widely found system in biological networks. So yeah. Uh, Rank of the matrix, I'm not sure how many of you like how many of you are aware of matrix algebra and everything. Just to give you a definition, the rank of a matrix is the number of, so there can be two ranks. One is the column rank and one is the row rank. When you are asked rank of a matrix, it's mostly column rank. We do not ask for row rank. Generally, it's column rank only. So you can assume they're asking for column rank. So column rank or the rank of a matrix is the number of independent columns. What does independent column mean? Can you do some, can you like combine these two columns, the first and the second to make the third, or vice versa, second and the third to make the first, something like this. So the columns need to be linearly independent. The number of linear independent columns of a matrix is called the rank of the matrix. In this case, you cannot make the fourth column, sorry, the third column using the first two. It is not possible. But say, for example, you want instead of the last column being 0012, if it had been 0, say 110 and 0. So this is 1, then 3 zeros, 0, 1, 2 zeros, and let's assume the fourth column was 1, 1, then 2 zeros. Then you could have made the third column as a summation of the first two columns. So then the third column does not remain linearly independent. So in that case, the rank of the matrix will be 2. But in this case, you cannot make one column using linear combinations of the other two. So in this case, the rank of the matrix will be 3. Right. So just the quick trick for finding rank of the matrix is if you can make one column, if you can produce one column as a linear combination of any other columns inside in the matrix, right? So if even if it would have been like 1, then 3 zeros, 0, 1, 2 zeros, and here it would have been, say, for example, 0, 2, 0, 0. In that case, if you multiply the second column with 2, you could have gotten the third column. So it doesn't remain a linear independent column. So in that case, the rank of the matrix would have been 2. In this case, it's 3. So that's the thing. Basics of matrix algebra you'll need. So if you are not familiar with matrix and determinants, please give a quick read. We'll not go into very detailed concepts of these things, but just to understand the terminology and what is happening, you'll probably need a little bit of matrix algebra. Okay. I hope I'm audible now. Yeah. Okay. Uh, do we have any answers? Yes. Okay. So to clear up a few things, so A, that that thing, that inverted comma thing, is basically A complement. Anything, if you write a matrix with a dash on top, what it means is A complement. Or sorry, A transpose. Sorry. I just confused complement with transpose. Transpose. So basically, if you have a 3 by 2 matrix, the transpose of that matrix is if you convert the rows to the columns and columns to the rows. So a 3 by 2 matrix transpose will be a 2 by 3 matrix. So matrix multiplication rule is you are if you are multiplying A matrix with B matrix, the number of columns of A must match the number of rows of B. So you can multiply a 3 by 2 matrix with a 2 by 3 matrix, right? But you cannot multiply a 3 by 2 matrix with a 3 by 2 matrix because the first matrix will have 2 columns and the second matrix will have 3 rows. So first matrix number of columns and second matrix number of rows should be equal for the multiplication to happen. So E and A complement can always be multiplied. So, sorry, I'm getting confused. A and A transpose, right? Its complement is different. A and A transpose can always be multiplied because if A is 3 by 2, the A transpose will be 2 by 3. So, always it is possible to multiply. Same with B transpose and B. You can always multiply a matrix with its transpose because the rows and the columns get interchanged. So, it can be done. Now, in this case, uh, A, A is 3 by 2. B transpose is 2 by 3 again. So A and B transpose can be multiplied as well. So all these three options are correct, except for the first option. The last three all can be multiplied. Yeah. Okay. Okay. The answer is 24 because it's MATLAB is one indexed, right? MATLAB doesn't start from zero. So when you're starting from k equal to 1, it's basically the first element of the B array. So it will add up 3, 8, 9, and 4. It's 24. What you did was add up 8, 9, 4, and 7, but MATLAB starts from 1. So it's 1, 3, 8, 9, 4. It will be added. So yeah, these kind of little things you need to keep in mind. But, yeah. Okay. Sorry. The answer is 24. So basically, when you are taking this loop, it becomes sum equal to sum plus b of 2 plus b of 3 plus b of 5 and b of 7. Because c of 1 is 2, c of 2 is 3, c of 3 is 5, and c of 4 is 7. So you take the second, third, fifth, and the seventh element of B matrix, B array, and then you add them up. It's 24. Okay, so let's move on to this. This is going to take some time, but yeah, let me know if you're being able to do it. Okay. Uh, okay. B. Yeah, I think it's correct. I don't know the answers here. Yeah, it's B. So just let me tell you how this works. So your A matrix, what it means is, so when it gives a semicolon, so means like 1 to 5 is the first row, 6 to 10 is the second row, 11 to 15 is the third, and 16 to 20 is the fourth. So what your matrix is a 5 by 4 matrix, sorry, 4 by 5 matrix. In the first row, it's 1, 2, 3, 4, 5. Second row, 6, 7, 8, 9, 10. Then third row 11, 12, 13, 14, 15, and fourth 16, 17, 18, 19, 20. So this is your A matrix. What your B matrix is, it's basically an array. It's a uh, it's an array which starts from 2, ends at 10, and there is a 2 increment of 2. So your B will be 2, 4, 6, 8, 10. So it's a 1 cross 5 matrix. So your A is a 4 cross 5, and your B is a 1 cross 5. Now you're asked to multiply A and B transpose. Your B was a 1 cross 5 matrix. Now if you take transpose, it will become 5 cross 1. So your A was already 4 cross 5, and now your B is 5 cross 1. Now if you this is multipliable because columns of the first matrix is equal to the row of the second matrix. You can multiply. When you multiply, you'll get multiply and add. You know how matrix multiplication works. If not, please read up matrix multiplication, and you'll get these. This is the result of that matrix basically. So yeah. So in the first row, you'll get 110. Second, you'll get 260. Third, 4110, and 560. So yeah, this is correct. Whoever answered it, good job. Yeah. The correct answer is 45. So basically, we have to take the unique elements in this particular array, which is basically 1, 2, 3, 4, 5, 6, 7, 8, 9, and just add up. It's 45. Yeah. Okay. So I hope you can see this. Okay. Answers. R is actually a coefficient of correlation. Basically, R is the uh, basically R square is given by it. Where did it? Yeah, it's R square. Basically, it's the percentage of variance explained by your x variables compared to your y variables. So R square, the greater the R square is R square equal to 1, then your model perfectly fits your data. Like it's a perfect fit. But it's not possible to have R square equal to 1. R square mostly lies between, if R square is greater than zero point 0.9, it's assumed that it's a very good fit. And if it is less than zero point 0.6, zero point 0.7, then it's not a very good fit. So R square determines the fit of your model. And like basically your correlation coefficient of your model. So if this is your curve, how much of your y can be explained by your values of x. So it's kind of a correlation coefficient. It tells you about the fit of your model to your data. So I forgot the answer. What should you do? Yeah, it's actually overfitting. If you see, it's like it's going through each and every data point. It actually should be around a straight line. It's not a sixth order polynomial model. It shouldn't be like that. So the model is definitely overfitting. So yeah. I have forgotten to put the answer here, but yeah, the model is overfitting, and therefore we can choose a bit better model even if the R square value is a little low. So, okay, I have put the answers yes anyway. So let me discuss this. This is, I think, okay. I have a couple of questions after this. So let me discuss this. So this is actually a toggle switch. So as I was talking about, so you have two elements which are repressing each other, and you have two other elements which are repressing these two. And the ultimate formation of GFP, which is the product here, this is also a part of a Lacko product. So that is produced by all four interacting elements. So you have a toggle switch. You have consisting of two promoters and um, and two repressors, and basically you have one-to-one connection. One promoter will one repressor will repress one promoter, the other repressor will repress other promoter. So, and there are two inducers, which is this L1 and L2. Basically, in this case, your LAC and lambda CL are basically your inducers, and there will be a particular protein that is getting formed across all these four elements, and then this is what becomes a biological circuit. So this is actually a network which interacts with each other to give a particular product. So uh, if these are the statements that are true for this particular case, so the presence of both IPTG and heat leads to partial GFP production. So you have IPTG and heat. So see what these are doing. IPTG is repressing formation of lambda Ci from lac I. So which lambda Ci will give GFP. So if lambda Ci is not present, it will not form GFP. Now same with heat. Heat is repressing lac Ci. Now, lac Ci on turn represses lambda Ci. So, if there is heat, the amount of lambda Ci is going to be more. So, you have two contradictory things here where there is IPTG, which is already reducing the amount of lambda Ci, and then you have heat, which actually through other things is increasing lambda Ci. So, there will be a partial production. It will not be completely halted, there will be a partial production. So, if you have only IPTG and not heat. So basically, your IPTG will be repressing lac I and this formation of lambda Ci from lac I. But if lambda Ci is not present, it's not getting formed. It's not being able to repress lac Ci. So basically, it's a cyclic mode. It's a complete negative feedback. But if IPTG is present, there is less of lambda Ci. There is less of lac I, which leads to more of lambda Ci. You're getting the point. So you have IPTG, less lambda Ci, less, more of lac I, and then it's completing the GSP production. I think I think there is some issue here in the answer. So basically, understand. So if your lambda CI is more, your GFP will be more. If how will your lambda CI be less? It will be less because of the presence of IPTG. But lambda CI itself represses LAC-I. So if LAC-I is less present, it will not be converted to lambda CI. So GFP will not be formed. The second option is actually not correct. It's a wrong answer. So that is why I think I put it like this. I've forgotten why I did this, but anyway, I think the second option is incorrect. I'll recheck and I'll discuss this question in the next class as well. Uh, I think it is wrong. And the third option, the presence of heat does not lead to only heat does not lead to GFP production is because you don't have IPTG here. You only have heat. Heat is repressing the formation of LAC-I from lambda CI. And LAC-I itself is not forming lambda CI again. So your lambda CI is very low, which in turn will not give you GFP. The first and third options are correct. The second option is I don't think is correct. I'll recheck and I'll discuss this in the next class. Okay. Any answers that you've given? Okay. Yeah, I'll recheck this question and I'll get back to you in the next class. This I don't think is correct. Okay, so before you answer the question, this is here is also a typo. This is basically going to be DSDT equal to uh, sorry, these three I is infected, R is recovered, R is not present here actually. So this is also a little bit of not exactly correct question, but anyway, let's go into it. Let's see. Okay, okay. Let me discuss this. So D is correct. C is correct as well. Huh, you have missed one option, which is the last one. Anyway, let me go through the model. So you have three sets of people, S, I, and R. S is susceptible, I is infected, R is recovered. Now, DSDT, DIDT, and again DSDT. So DSDT is basically the rate of change of the number of susceptible people. How will the rate change? It will depend on the number of susceptible people also, and also the number of infected individuals. Same with in the last equation, it only depends on the infected individuals. But these two, DSTT and DSTT is explained by these two phenomena. What I think is there is a typo here. This should be an R. The rate of recovery would also depend on the rate of infected people. But anyway, let's assume what is given here only. So your DSTT is dependent on S and I, and here it is I. So your susceptible number of people is dependent on the number of people that are infected and also the number of people that are susceptible. Now you have the infected people, DIDT, the rate of change of infected people, which is dependent on the number of infected people and also the number of susceptible people. And the first option is incorrect and the second option is correct because your N remains constant throughout is DSDT and DIDT. So your n is not changing. So your n is constant, which is the total population is constant. And there are only three categories of people that is given anyway given here, S and I and R. The recovered people cannot be infected because this is the rate of infected people that rate of change of infected people, DIDT. Here is no mention of R. If R would have been present here with a parameter, then you could say like the recovered people are also getting infected. But in this case, the rate of change of infected people does not include the recovered people. So it does not depend on R as well. So the last option is also correct, in addition to B and C. The fourth option, the infected people would not be susceptible. That is wrong because the susceptible people actually are dependent on the number of infected people. So it depends. Infected people can infect the susceptible. So yeah, I think this is the last question for today. Yeah. So basically, this is the ODE that is given, and the curve is made from these ODEs. Just to be clear. Okay. We have answered. Sorry, I did not notice this. No, the thing is, I assume that the question is correct, though I feel it is wrong. The equations are wrong. So the recovered people, there is no across the three equations, there is no mention of recovered people. So the rate of change of susceptible people or the rate of change of infected people does not depend on R. That is what the logic that I am using, because there is no R in the equations anywhere. So it does not depend on R. Both DSDT and DIDT. So that is why the recovered people cannot be infected. If it is like DRDT equal to gamma I, if that would have been the case, then I would say like obviously the recovered people can be infected because number of recoveries are depending on the number of infections that are present. So that would have been true. But here we are assuming this equation is correct. Uh, Ha, your understanding is correct. So if it would have been DRDT equal to gamma I, then obviously they would have been susceptible. Yeah, true. Okay. Do you have any answers? I hope you understood that. Yeah, you're correct. So if it would have been a typo, then recovered people would have been susceptible. Okay. So let's discuss this. So basically, it's the question looks very big, but it's actually a very simple question that they're asking. This actually this question is like the information given in the question is very important because you see this curves that you see S, I, and R curves. This is how in during the pandemic even we used to predict how much population percent of the population is susceptible, how much of it has been recovered, and everything. Technically, the flattening of the curve means it's the reduction of this I curve, basically. We obviously with this reduction, the recovery rate also goes up. It's complementary, so it will go up. And if the susceptibility, if there is a reduction in infections as well, the susceptibility rate will also go down. So the question is like how what is flattening of the curve? The flattening of the curve is the rate of infection that is going down. But this is actually how a curve, an SIR model looks like. So there are various nuances to this SIR model. You'll see different variations to SIR model. If you do a Google search, there will be SIR. There will be SIER. There will be SIRV. You add an element of vaccination and what it is doing to that to the pandemic as well. So there can be various variations of the SIR model, but the SIR model is the most basic one. We will study the SIR model, I think, in the next class, if I'm not mistaken. I've forgotten. I have to check. But at some point of time, we will study the SIR model, and it's actually a very useful and a very uh, it was very widely used during the pandemic to predict a lot of things. So yeah, so that's all from me for today. If you have any questions, you can ask, or we'll close the session. Okay, guys, thank you. And I'll leave my email ID here if anybody like anybody has any questions or anything. Uh, or if you want to if you want me to discuss anything in particular for the next classes and everything, you can like email me prior to the class, and I'll try to cover those topics in these sessions. So, okay. Please call me Shomu, don't call me sir. Okay. So I'll drop off and close this session. Thank you guys for joining. See you next week, Tuesday, same time. Bye bye.